what do we understand from the term drugability? So what is drugability? As the name suggests, it is basically property of a molecule, which is of course protein, to have good chances of becoming a drug. Now, what are the mechanisms and where we stand at current times in drugability scenarios? So if I start from the post-genomic area, basically, which is basically once we have information related to genes of different organisms with us, such as we have Human Genome Project, in which we have information about different genes related to humans alone. So in this post-genomic era, what has happened? That we have information about different genes, then different proteins, gene and protein expressions, protein-protein interaction, and so on. So as this information or literature about different macromolecules increases, there is good chances of discovering new drugs also. Now what is the setback basically? Setback is again that although we have very huge amount of different molecules available, but still we don't have a good number of druggable molecules. So we want to move towards that molecules which can be used for drugs also. So if we say that we want to move towards drugs or targets which can be used for drugs, then we need to have a target validation mechanism. Now what is target validation? Basically, when we talk about target validation, what we know about targets first? We should know that targets are basically those macromolecules which are associated with diseases. So association with disease means that they can be used for generating some disease, then progressing of this disease, and then further they can be used for cure or treatment of that disease. So for that purpose, what we have to identify different drug targets which can be used or which can be done by gene expression profiles. And finally, what we can say that drug targets can be manipulated by outcome and progression of phenotype organisms. So in order to solve these target validation, we have different mechanisms available. First mechanism is gene type variations or gene dependent initiatives. So gene dependent initiatives would be in which we are using genes information for remedying drugs, for remedying diseases, or for making drug targets. Now what happens in this information is we have to understand two types of disease genes or proteins. One is disease associated and the other one is disease modifying genes. So what are disease associated, which cause diseases, and disease modifying, which remove diseases or which provide treatment of against the diseases. So there are two methods available, as we have seen in different modules also, that one approach is forward genetic approach and the other one is reverse genetic approach. In one approach, what we do, we have some disease in a phenotype behavior and then we try to find out what is its effect on the genotype thing. And on the other hand, other approach, what we have reverse approach, that in which we have information about genetic information and then we try to find out its effects in phenotype based approach. Then we have used the word mutagenesis. Mutagenesis are basically those animals which are used in vivo setting, in laboratory settings, for human testing type things, such as if we want to apply some drugs on humans, we first try this drugs on some mouse or rat. And how it is done, that we first put some disease or those genes which are causing that disease inside the rat or mouse, and then we try to cure those diseases. Then about protein drugability. Now what about protein drugability? When we talk about protein drugability from Human Genome Project, we already know that there are 3000 gene loci which are available for finding druggable proteins. Now how we come to this 3000 count? Basically this 3000 count is taken by different databases, gene expressions, gene profiles, biological sequence literature, and so on. So when we have got this thing, we have two more databases which are Interpro and PFAM, which provide us information about different protein sequences. And finally, if we have such a huge amount of data available, best approach to solve or to use this information is computational approach. Why? Because computer can handle large amount of data in quick time intervals to solve optimal problems. So now if I want to define what is a druggable protein, how I will define a druggable protein? That is defined as that a protein which can bind small drug-like compounds with high affinity and specificity. So what are affinity? Affinity means that we have chances that a drug-like molecule, such which is basically a ligand, it can bind to a protein, and specific means that it should be specific to that protein alone. So what will happen? To find out these two things, we should have structural information of the given protein set. So what will happen? That modulation of biological function may be suitable for therapeutic invention or may not be suitable. So what does this mean? In some molecules, what happen? We already know that they are very good or very important with respect to biological information. But are they druggable? That is a question mark. So for some molecules, we know that they are good in drug making, but their biological importance is unknown. So when we talk about or when we want to make medicines, we should be sure that this macromolecule is good both biologically and for drug making mechanisms. So how we say that what are the probable binding sites which are available in a given protein? For that purpose, we have two options available. Number one are geometry based approaches, and the other one is so what are geometric based approach? Basically, geometric based approaches are those approaches which make use of the geometric structure of the protein. Geometric structure means that we have as we say that when we try to bind to molecules, so what should be there? That the bigger molecule should be concave in nature. So concave nature means that it would have larger chances to have or to mix the outer molecule, which is small molecules in our case, that they should combine in the same place. And then there should be a mechanism of specificity. So specific means that a specific ligand should be binding to a specific protein. Other part of this probable finding sites can be done by using energy based mechanisms. In energy based mechanisms, what we have basically, instead of geometric based, which are based on surface or concave type things, energy based mechanisms are based on information which is being used by taking the energy of the molecules. So what is those energy based molecules? That sites which have higher energy level as compared to other sites, they can be used for binding ligand with the protein. So now what we conclude from this all this module is that for binding a ligand with the protein, we have to take care of two things. Number one is its geometry and the other one is its energy. So, and what are the two parameters? Those parameters are, number one is affinity. Affinity means that the binding position, that what are the, if it is concave, then how much it has affinity position, how much position is available or location is available for that binding. And the other one is specificity, and specificity means that those molecules should be specific to each other.